Very good afternoon to everyone. Are you able to hear me? Anybody can respond, please.
Yes, sir. Yes, sir.
Oh, okay. Very good afternoon to one and all. On behalf of our director, he is having another important meeting at NBPGR. I extend a warm welcome to all the participants, both online and offline, for this today's webinar. This is the 22nd in the series of seminars organized by Indian Institute of Rice Research Seminar Committee, as well as Society for Advancement of Rice Research, as a part of Azad ka Amrut Mahotsava celebrations. On behalf of this society, as well as Indian Institute of Rice Research, I extend a warm welcome to our beloved guest speaker, Dr. Habil Joerg Fettke, Head Biopolymer Analytics, University of Potsdam, Institute of Biochemistry and Biology. He's from Germany. So thank you very much, sir, for giving your time. Today, he will be enlightening us on new aspects of starch metabolism, insight by looking closely. As you all know that this is very important. Starch is manufactured in the green leaves of plants from excess glucose produced during photosynthesis and serves the plant as a reserve food supply. Hence, it is very important to understand the dynamics of this important energy source. Once again, I thank our speaker for taking his time to come over here and deliver this lecture. To start with, I request Dr. C.N. Neeraja Madam, who is Head of Biotechnology, to introduce our eminent speaker to our audience. Thank you, Madam. Thank you.
Hello. Hello. Are you able to hear me? No.
Yes, ma'am. Yes, ma'am.
Yeah. Good afternoon. Divisions, improvement. Why is it? Okay. But I think I'll continue. So, Dr. York is from University of Potsdam, Institute of Biology and Biochemistry and Biopolymer Analytics from Potsdam, Germany. He has completed his diploma in biology with honors and PhD thesis and a PD in plant physiology. Now he is professor in molecular plant physiology. He has supervised about 14 PhD students, 5 masters and 4 diploma students and 17 bachelor theses. He is an editor in academic editor of PLOS ONE, and he reviews many projects of national and international and European projects for their funding, and he is also a reviewer of many reputed journals like Trends in Plant Science, Plant Cell, Plant Physiology, and so many journals with high impact factor. So as he has been a leader of mass spectra for of biopolymers at University of Potsdam. Later on, he is project leader of signals sensing and plant primary metabolism at the same university, and that later on PI group plant physiology, then acting professor and visiting professor to University of Life Science Warsaw, Poland. Now he is leader group of Biopolymer Analytics, and he has many publications in very reputed journals, international journals with high impact factor, especially, and his specialization is starch metabolism. We are looking forward for his now presentation to give a direction or suggestions for which we can do in rice starch metabolism, which will impact quality and yield in rice. So thank you, sir. Thank you for giving me this opportunity. So I invite Dr. York to give his seminar today for all our SARR members as well as ICAR partners and our institute research group. As I welcome Dr. Dr. York.
Okay, thanks a lot. I hope you can hear me. Parana, give me a sign if you can hear me. Okay. Okay, what first of all, I learned already that I have to speak a little bit slower because I have a different English, and so you have maybe then easier to understand me. And we also have already a discussion with members of the institute, and so we also decided to change a little bit. So I will give an overview, and then Julia, one of my group members, will a little bit focus on the analytic, because that is something you not do here, and so you can have a better idea what we exactly do in our analytic research. So just to give you a short idea about our group or my group, as you can see, it's a relatively international group, and we are localized in Potsdam, as already mentioned. Some of you I know already know Potsdam. It's a really close city to Berlin, and it's a hotspot of plant science in Germany. So we are very close to the Max Planck Institute and the Fraunhofer Institute. We have so a very good environment for research. And as you already realized, so the group has a name, Biopolymer Analytics. So we are really focusing on analytics. That's why we have a really good infrastructure. So we have different mass spectrometry devices, we have chromatography devices, multi-sizer, capillary electrophoresis, and so on. And we also are able to do all these experiments radioactive, but it's very important to because of the sensitivity, as we are really looking in small alterations which have a really big impact. You can easily think about, for example, post-translational modifications, and their radioactive tracing helps a lot, but also for flux analysis to understand how metabolic pathway happened in plant. And our main research interest is carbohydrate metabolism, and here especially starch metabolism, but we are also interested in the link between carbohydrate and lipid metabolism. And as I am a guest here on this institute, which is focusing mainly on rice, I also want to give you an idea about our main models. Our main models is potato and Arabidopsis thayana, the typical molecular experimental plant. But in addition, as you can see, we also work in oil palm, and in some cases we also work in algae. And our group is really shows a broad spectrum of experimental approaches. So we combine biochemically, biophysical, molecular biology, and plant physiology approaches to really understand what happened in the plant with the metabolism. I skip that because that is not so interesting, because I want to short it up so that Julia has also time to present. What you can see is this is a typical starch pathway already known in the last years in Arabidopsis. And what you can see clearly is that we have a lot of enzymes which are involved. We have a lot of glucans which are involved, and we also have different pathways, and we have transporters. And what is also surprising is that the starch metabolism not only takes place in the chloroplast or in a amyloplast, if we go to storage starch, it also has a cytosolic path. And surprisingly, this cytosolic part of starch metabolism has a really huge impact also on the starch metabolism in a chloroplast itself. And I will show you very short examples for that. I will not go too much into detail because what I present here is mainly an Arabidopsis, our model system, but just to give you an idea what we're doing. And then, as I mentioned, we switch to you to show you the analytics. And if you go to transitory starch, and especially transitory starch degradation, it is really surprising that starch metabolism or transitory starch metabolism is really mostly active in Europe. So it's a really unique set in contrast to other research where the US is really dominant. Western Europe is on a top level on this research. And now coming to the idea, what we are trying to understand in starch metabolism is, as you can see, starch has a really big impact. So we don't only have to think about starch in feeding or food. You have a lot of applications of starch. And this is, for example, biofuels, but also biomaterials, paper industry, glue industry. So a lot of different applications. And that's why you have to think about two totally different perspectives. One is the amount. Of course, it's interesting to have a lot of starch. So you want to increase the yield, like here's a farmer in Germany with this really huge tuber. And on the other side, there is a starch property. And this starch property, the starch properties are different parameters which can or which should be recognized. And here in the down, you see some examples. So the starch granule size can be totally different. The starch granule morphology can totally be different. And other parameters can also be different. I come to this later. And you can easily imagine that the application of this starch depend on these parameters, because if you want to produce a glue, you have to have small granules which show strong interaction to each other. And if you, for example, want to use it in a paper industry, you want to have no sticky starches. And that's why if you understand how these different parameters of a starch granule can be affected, you can easily adapt that for the application. And that is what we are mainly interested to understand. And here's another example. So what you normally do also in case of rice is that you isolate the starch from the plant. And after this isolation of starch, after the isolation from the plant, the starch has to be processed. And what you can see here are 4 easy examples of starch processing. And you can see the source of the starch strongly affect the necessary processing. You can see here in case of pea starch, you need a really low step protocol for processing in contrast to potato starch or wheat starch. And this is coming back to the idea I already mentioned before. So if you can influence the parameters, you can influence this processing and so make it easier for application. These parameters I mentioned here, the size, the morphology, and also the inner structure, are in the moment not understood how it exactly is generated. What we know is here typically depicted as a starch structure. You have mainly amylopectin molecules. In addition, you have amylose, and the amylopectin molecules form a specific structure. As you can see here, I don't know if you can see my mouse. As you can see, so it's mainly linear alpha-1,4 linked glucose units, so glucosyl residues, which are interconnected. And in addition, you have clustered alpha-1,6 linkages. And this clustering is very important because that makes a difference to glycogen, because here they are organized in a clustered way, which in principle allow this specific formation of double helices, which is typical in an amylopectin molecule. And in addition, the organization of this different double helices can be again in two types organized. One is the so-called A and B type. So what does it mean? As you can see, they are differ in the water content. We call that allomorph because that is physically determined. And you have to keep in mind, until now, we do not understand how this clustered structure is formed. We do not understand what defines the allomorphs. Yeah, so it can, in my opinion, not be just related to biochemistry. That is also physically driven, how this is organized. And we want also to understand that. Overall, it seems that the inner structure is somehow conserved. From all known starches, we found this conserved structure. And the second part of, or the second molecule inside our starch granule is amylose. Amylose is essentially alpha-1,4 linked. We have nearly no branches. There are some descriptions that also amylose contain a few amount of branches, but the idea today is that it's mainly a linear molecule, but we also do not understood where it is really localized. Sorry. You see here two main difference. One is this orientation through the double helices, and the other one is inside the amorphous region. We don't know exactly where it is, but of course that can massively influence the properties of a starch molecule or starch granule. And coming back to the metabolism, here we give you an overview what happened on the phosphate surface, or the phosphorylation on the starch granule surface, as this is the only covalently known modification of starch, and it has a really huge impact on the starch structure and metabolism. And I want to give you one example about the connection between the different parameters. So what are the different starch granule parameters? First of all, it is the number. You can easily imagine if you generate a lot of granules or a low amount of granules, that has a massive influence. In principle, that is also strongly connected to the question of initiation. Today, we don't understood how the starch granules are initiated. We don't know how the number is regulated. We make some progress, so we had a lot of Newton which help us to understand this, but it's not known. Similar morphology. The morphology, how the different surface morphology, is it round, is it flat and discrete, how this is regulated. We do not totally understood. We have again some mutants there which help us to understand that. Same the size, and you connected with the size.
Here only, no?
Sorry?
Maybe it is disturbance, you can continue.
Okay. The size, and you have to keep in mind we speak about carbohydrates which are not determined genetically. So we have not a one size, we have a size distribution, and how this size distribution is generated and regulated, we also don't know. I mentioned already the inner structure, and if we come to the last, then it's a surface. The surface, you have to keep in mind, is an part of the insoluble starch granule with the water area. And so it's this surface is where the biochemistry take place. So where all the enzymes act during synthesis and degradation. And so it's also really important to understand exactly what happened on this surface and how this surface property influenced both the synthesis and degradation, so the starch metabolism. And if you think about first about this parameters, you can easily ask questions like, are they connected? So if you change the morphology, will you change the size or not? Can we separate them? And also, are they steady? So if starch sizes are stable over the development or the development of the plant, or are they alter during aging, for example? And I will give you a short overview of that. I skipped this part. What is important already by mutants, we learned that we can, in principle, cluster them. So we can find enzymes which somehow influence the initiation. We can identify enzymes which are somehow alter the structure and the size, and we can also group them in enzymes related to synthesis, others to degradation. But in the end, what is a big problem is that the mutants are limiting, because, as I already mentioned, if we speak about starch, it's not genetically determined, it's metabolically regulated. So you have to always to keep in mind that you have carbohydrate fluxes, and you have in fluxes and out fluxes of the starch granules. And that's why if you just use mutants and knock out one specific plant, other pathways can take over this activity, or other enzymes can take over this action, and that's make it more complex. And what I want to show you very fast is an example of a double mutant. As you can see, and I already mentioned that, this is a double mutant. One of the enzymes is localized in a chloroplast, so it's called PHS1, so it's a plastidia phosphorylase, and the other one is in the cytosol, so it is not, or has not a direct access to the starch granule because it's outside of the chloroplast. And what you can easily see is, if we can prove that the activity is gone, so you here have native pages, so the activity is staining, and you see DP2 activity is missing here, the PHS1 activity is missing here. And if you look then to this double mutant, you can see it has a really tiny growth. So the plants are really tiny in comparison to the wild type and also to the parental lines. And what was surprising, and I want to speak about starch today, you see starch can be easily stained by iodine, and this forms a black color if you have starch inside the leaves, like here in the parental line DPE2. But the double mutant show no starch in most of the leaves. Only in the very young and tiny leaves you have some black color which reflects starch. So it was surprising why the starch distribution is altered in this double mutant. And coming back to the other question, the granule number. So we had a closer look to the starch granule number in these mutants. And on the top, you see the typical parental lines, and you have normally 5 to 7 starch granules per chloroplast in these lines, as you can see. But the double mutant DPE2 and PHS1, as you can see, shows either no granules in the big leaves or in the tiny leaves. As you remember, these tiny black leaves, you find one starch granule, which was spherical and really huge. So the double mutant is not only affected in the starch distribution in the leaves, it is also affected in the starch granule number regulation. And of course, we want to understand the physiological impact. So we asked also the question, what happened if we put the plants in continuous lights? What is the background idea of that is, under continuous light, you don't have any starch degradation. You have only starch synthesis, because we speak about transitory starch. And what you can see here, the growth phenotype is more similar to the wild type and the parental lines. And also, what is surprising, the starch amount is really huge, and the starch distribution is normal. You also see starch in the big leaves. So what does it mean? That means that under continuous light, the starch distribution and the starch amount is totally different regulated than if you have a typical dark light change during the growth. Okay, we also are interested in understand the entire carbohydrate metabolism. So we make metabolic profiling, and what is the outcome I want to show you here very fast. Here is a double mutant in the typical light-dark regime, and you can see all metabolites we analyzed here are strongly affected. In contrast, if you go to continuous light, the double mutant is here. It's more similar to the wild type and the parental situation. And that is exactly what we expected from the continuous light, from the growth characteristics, and also from the distribution of the starch in the leaves. On the right side, you see we can also show that by a different second mutant, MEX1 PHS1. MEX1 PHS1 is the same pathways are affected, but MEX1 is a little bit upstream of DP2. It is a transporter of the maltose, and we see here the same phenotype. So it seems that what we see is related to two pathways during starch degradation. And you have to keep in mind, starch degradation seems to affect the starch initiation, the control of the starch granule number and the granule size. Okay, what I showed you is under continuous light, this effect of the reduced granule number was not any longer there. As you can see, under continuous light, we have the typical number of starch granule. And that also points to the next question. What is happening if you omit the dark phase? In principle, two options exist. One is you need the dark phase per se. You need a starch degradation. And the starch degradation is mainly controlled, again, by a phosphorylation, dephosphorylation cycle on the surface. Julia will speak about that, as I mentioned already. So what is important to know, I only want to go very short. You have two dikinases, and they phosphorylate the glucans. Julia will explain in more detail. And what is important is one of these dikinases, the GWD dikinases, is really effective in controlling starch degradation. So if you knock out GWD, as you can see here, there is no starch degradation. As a reminder, we make here iodine staining. So if you have a starch inside, it's black. And we put them under longer darkness phases. And you can see in the wild type situation, after 12 hours darkness, the starch is used up. But in GWD, even after 84 hours of darkness, starch is not degraded. So GWD totally blocks starch degradation. And as you can see, the PWD mutant, this is the Azad dikinase. Here you can see it is delaying the starch degradation, but starch degradation is still going on. Okay, so to test now if our starch degradation is essential for what we see in continuous light, we generate a triple mutant. So again, PHS1, DP2, our double mutant, and we introduce SEX18, which is the GWD protein. And as you can see, again, here from native pages, from activity staining, we have no DP2 activity, we have no PHS1 activity, and GWD is missing in the line. So this triple mutants, as you have a look to them, grow better than the double mutant, has higher starch amount than the double mutant, and you can see the distribution of the starch is totally different again to the double mutant. Yeah, so it is that you can somehow rescue this starch phenotype. So that means that really the starch degradation is important for what we observed there. And you can, in addition, also have a look to the granule number, and you see in the triple mutants, the starch granule number is increased again. So that means starch degradation and not darkness per se is important. We also did that, as I mentioned, for the PWD mutant, which still shows a little bit degradation. So you have not a total block. And it's not surprising we get an intermediate situation. In the triple mutants with PWD, you have an increase of the starch granule number, as you can see. So a low amount of ongoing starch degradation results in an intermediate situation. So it is a good proof of concept. We also do here phenotypic characterization. I don't want to go in details to that here. And we asked then the question, if this regulation is stable. And so we looked a little bit closer to the growth phenotype. And here you can see the comparison of the leaf numbers and also the fresh weight in the wild type situation and in our double mutant situation. And what you can see is there is a phase where the growth is reduced. Here you see it's the same. So the fresh weight is reduced. And after this phase, the growth restarts. So you have a phase where it grows normal. Then you have a delay phase. And then you have the situation that the plants grow similar again. And we call these periods, as you can see here, the prior period, the recovery period. And this is a reduced granule number period I mentioned before, where we see this one granule. So that means the plant growth and also the starch metabolism are not stable throughout the plant development. So you can clearly distinguish three phases. And I skipped that. And here, if you look to the granule number, you can again see this is the wild type situation, the granule distribution. The prior period, you have a relatively simple granule number distribution like in the wild type situation. Here is the reduced granule number. As I mentioned, mainly one in young leaves, in old leaves, mainly 0. And after that, the recovery period, you come to a situation which is again more similar to the wild type situation. That means also here again, you have a clear impact that during plant growth, the number regulation is affected. We also make transcriptomic analysis and we're able to identify different clusters for different starch-related enzymes. That is what we're working in a moment on to understand which enzymes are really affecting the situation in this double mutant during the aging. You can easily imagine that you have very good clusters where, for example, in the reduced granule number period, all the transcripts go down, or like here where they go up again after this period. But this is something we still work on. Coming to the next, parameter, the inner starch structure and the starch morphology. And you can see also here in these different periods, the morphology is altered. So they come from more spherical to a round shape and then back to a more discoid shape. And this is also accompanied by changes in the inner starch structure, so the amylopectin structure, so the chain length distribution of the amylopectin molecule. And this is also really interesting because here it looks that this is interconnected. And we ask the question, is that typical for all different or for a lot of different mutants? And so we screened other mutants for that, and we found no, not all mutants show alterations. Some show changes with the age in the diameter. Here you can see the maximum diameter and the minimum diameter, they are affected. But other lines, as for example SS4, show no alteration in the diameter. So not all mutants are affected in the starch granule size or morphology. We were also able to break that down to specific enzymes which are involved in that. I want to skip that. And what you can see again, if we stay with this mutant I mentioned before, here's the DP2SS4, they show this effect, and SS4 not. And if you now go to the inner starch structure, you can see SS4 also shows no alteration in the inner starch structure during aging. On the other side, the other mutants, which show alteration in the morphology, also show alterations in the inner structure. That means the change of the morphology is accompanied by a change of the inner structure. That I want to skip today. So, coming to an end, what we know, what I try to show you is that we miss still a lot of understanding how this starch parameters are controlled. And I showed you that some are connected and some not. And we want in the end to understand exactly how we can influence this different starch parameters. And I want to, in more general, again, mention we have to keep in mind that these parameters are metabolically determined, not per se genetically. That you can easily see if you look at the double mutant, that it changed during age, the number of the granules, the size of the granules, the morphology of the granules, and the distribution inside the leaves. And I showed you that this is also the case for a lot of other mutants. So it is really interesting to find out that, but you see, it's a lot to do there. And with that, I want to stop here and acknowledge the people who work on this. And especially, I want to thank Xingting Lu and Xiaoping Li. These two Chinese really work on that, what I presented today most. And now I want to thank you for your attention. I'm not sure if we should make now questions or if first Julia should show her presentation. I cannot hear anybody, so that's why I...
Hello, sir. Am I audible?
Yeah.
Sir, let us continue with Julia's presentation. At the end, we will take up all the questions.
Okay. Okay. Thanks.
Yeah, no. You can just.
No, I can do it. I know. So.
Thank you. Hello. Today I'd like to give you a short overview about the starch phosphorylation, impact, challenges, and analytics. First of all, I can skip.
Next.
Sorry. Great. Okay, sorry. Okay, first of all, I'd like to give you a short overview about the history. In the beginning of the 70s, they discovered the phosphoesterification of starch. It is the only known covalent modification of starch until today. Almost 30 years later, they discovered a granular associated protein, which they called R1. And 4 years later, they discovered the function of this enzyme. It is a dikinase, and they called it alpha-glucan water dikinase. 3 years later, 2005, they discovered another dikinase, the phosphoglucan water dikinase. And then 2006, they discovered that GWD and PWD catalyze different phosphoesters. What does it mean? It means GWD phosphorylate only the C6 position within the glucan chain, and PWD phosphorylate only the C3 position within the glucan chain. Here on the here you see the mechanism of the reaction of the dikinases. The dikinases use ATP as a phosphodonor. They take the gamma phosphate to transfer to the transfer to water, and then they take the beta phosphate to transfer it first to the histidine residue within the catalytic center, and from there to the glucan chain. And what I mentioned before, the reaction of the dikinase is the same, but GWD phosphorylate exclusively the C6 position and PWD only the C3 position. Here I only want to show how important the phosphorylation is, and it was also explained by Professor Fettke already. But what you can see on the left side is when you block the phosphorylation by GWD and PWD, the whole degradation pathway is blocked. So how the dikinases are regulated? Some years ago, we find, and also another group find two proteins. They called them early starvation 1 and like early starvation 1. And what is really interesting is that when you look to the wild type here on this side, is that the wild type normally synthesizes starch over the day, and then it degrades it normally. But the ESV1 mutant and also the LESV mutant show that the normal starch synthesis over the day, but the consumption of starch is much faster in comparison to the wild type. So it means they start starving during the night, which influence all, of course, the growth. Here you see the comparison of the protein domains. ESV1 and LESV has a homology of around 40%. And the biggest homology they have in the tryptophan-rich region, which is predicted to be a carbohydrate binding domain. The difference between ESV1 and LESV is that ESV1 has a protein-rich region, but the function is not known until now. So we know they start starving during the night, the ESV1 mutant and LESV mutant. So we tested in vitro the impact of ESV1 and LESV on the dikinases. Here in the first row, you see the impact on ESV1 on GWD. On the left side, the impact of LESV on the GWD action. And for both proteins, the phosphate incorporation by GW is inhibited, or it is reduced. So it means in presence of ESV1 or LESV, GWD is not able to incorporate the same amount of phosphate as without. The same is for PWD, or the same you can see for PWD. PWD can incorporate less phosphate in the presence of ESV1 or LESV. But what is really interesting is when you pre-phosphorylate the starch by GWD, and afterwards you incubate it by PWD with LESV or ESV1, you see an opposite effect. You have more incorporation of phosphate. Sorry. So there are so many applications for phosphoesterification of starch. Why? The phosphate groups changes the properties. So it decreases the crystallinity of the starch granules, it decreases the retrogradation, but it increases the swelling power and also the shear stability. You can phosphorylate the starch either in vitro with recombinant enzymes, what I'm doing, what I show you later, or by chemicals, and then you can use it in industry for wastewater treatment, textile industry, or paper production, or also for food production. What you can also do is you can do it in vivo by biotechnological tools. So you can overexpress, for example, the dikinases, or you can reduce the expression of the dikinases to change the granular properties. So the aim of my study is now to find out if GWD has a phosphorylation pattern. What does it mean? Does GWD only phosphorylate the surface of the granule, starch granule, or does it only phosphorylate it close to the branching point? Or if you have several phosphorylations within one chain, it can be also that every second glucoside residue is phosphorylated, or it is randomly, or maybe only one, or every third glucoside is phosphorylated. So another question arises if ESV1 or LESV have an impact on this phosphorylation pattern, and if the phosphorylation pattern is for different GWDs same or different. So now I'd like to show you my workflow to analyze the phosphorylation pattern. First of all, I phosphorylate the starch by GWD in vitro. Next step is I treat the phosphorylated starch with isoamylase, because isoamylase cuts the alpha-1 6 linkages. So we release the branched glucans. In the next step, we use thin layer chromatography to check if the phosphorylation worked and also the isoamylase treatment worked. If so, we go on and we do anion exchange chromatography. Why? Because we have a lot of neutral chains still, and this neutral chains will interfere with the analysis later. So to check if the anion exchange chromatography was successful, so we also do a thin layer chromatography. If it was successful, so we do a beta amylase treatment. Why beta amylase treatment? Because the beta amylase releases maltose units from the non-reducing end until it reaches a phosphate group, so it stops. And to compare the phosphorylated glucan chains only treated by isoamylase and compared with the glucan chains treated by isoamylase and beta amylase, you can estimate the position of the phosphate group within the glucan chain. After the beta amylase treatment, we analyze this by mass spectrometry or tandem mass spectrometry. So here's one example for mice amylose extender starch. On the left side, you see the thin layer chromatography for after the isoamylase digest and after the anion exchange chromatography. And on the top, the spots here are our phosphoglucans. So on the right side, you see a spectra of single phosphorylated glucan chain, of the distribution of single phosphorylated glucan chain. The blue one shows the glucan chains only treated by isoamylase, and the black one shows the treatment with isoamylase and beta amylase. And what you can clearly see is there's a shift. So it means that GWD never phosphorylates the granular surface. It always penetrates the granular surface a bit to attach the phosphate group. I have also done it for several other starches, also for maize waxy or Arabidopsis wild-type starch, and also for potato wild-type starch. And if you look to the distribution after isoamylase and beta amylase treatment, the distribution is similar. And what is also obvious is that for all starch types, you have this shift. So it means that always GWD penetrates the starch granular surface. It never phosphorylates immediately the surface, it always penetrates it. So what you can also see is that GW never phosphorylates the same position within the glucan chain. Otherwise, we wouldn't see this distribution, this Gaussian distribution, which you can see here or here or here. So this is a normal fragmentation pattern of a single phosphorylated glucan chain. I use DP6, which is depicted here as an example. And what you can see is that all different DPs can be phosphorylated. It means the phosphate group can be at the DP6, DP5, DP4, and so on and so on. And what is most interesting in this regards is that also the DP1 with the reducing end can be phosphorylated. So it means really that GWD goes into the granule and penetrates the granule and phosphorylated next to the branching point. So then we have also another dikinase in our lab from oil palm. So we wanted to know if they have the same action mode. So we compared potato GWD with oil palm GWD, and what you can see for different starch types, they have the same mode of action, but for maize amylose extender, it's different because you see the potato GWD incorporate much more phosphate than the oil palm GWD. So the question arises, okay, when there's a different action mode, maybe there's also a different distribution pattern. So I checked this. So you can see it here. On the top are the starches treated by oil palm GWD, and here you see the starches treated by potato GWD. And what is clearly visible is that it is completely the same. This is the same distribution pattern. But why then we have here this difference? Why oil palm GWD incorporate less phosphate than the potato GWD. The hypothesis is that the structural properties of mice amylose extender reduce the phosphate incorporation by oil palm GWD. But it doesn't influence the location of the phosphate group within the glucan chain. So now we come to the outlook. Of course, we know that there are also exist double phosphate glucan chains. So we want to know how the pattern looks like. And also we want to know the phosphorylation pattern of PWD from different species. And also how is the impact of ESV1 and LESV on the phosphorylation pattern. And another question that also arises, maybe the dikinases are able to use different substrates than ATP. Now I want to thank you. And now I want to say thank you to Professor Dr. Joerg Fettke and also to Ada Aprianto and Akansha Singh. And of course, also to Harindra Mahato. Thank you.
Nice. For questions.
Yes.
Hello,
Hello,
am I audible?
we can hear you.
Yes, yes. First of all, I thank Dr. Fettke for his wonderful lecture on the structural aspects of starch metabolism, and he stressed on the dynamics of starch granule in double mutants and triple mutants in relation to PHS and PWD enzymes. And next, he also stressed on the DPE mutants and SS4 mutants on the alteration in starch structure. Although this aspect is very, very hard to our colleagues who are basically different, they are different from biochemistry. Rarely we see this kind of research, and they do only basic research and applied research. And in basic research, we only do the peripheral aspects. And therefore, I hope most of our colleagues got inspired by listening to this lecture. And Julia stressed the new aspect of starch, that is phosphorylation of starch, and the mechanism of starch phosphorylation, and the two enzymes that are involved in starch phosphorylation, GWD and PWD, and their specificities in linking phosphorus to C3 and C6 positions, and the manner in which the starch is phosphorylated in the starch molecule during its growth. And they also explained the manner in which whether the phosphorylation takes place at the periphery of the starch granule or the internal aspects of the starch granule. And although this is still this research is still ongoing, as per the lecture, it indicates that the phosphorylation happens at the periphery, and she also showed with the help of mutagenesis and also isomerase and isomerase treatment, the variation in degree of polymerization of chains in the amylopectin molecule. And we thank both of you for a wonderful lecture. And our institute is also thankful to you for taking your time and coming to us all the way from the Germany with your team, and giving this lecture. Now the floor is open for any questions, and I request, if anyone is interested to interact with the speakers, please raise your hands, or else type your questions in the chat box, so that it will be more convenient for them to go through the questions and then answer, because our pronunciation is different from their pronunciation. I think I request most of the people to enter your questions in the chat box, so that they can easily interact with us. Thank you. Anyone wants to interact with the speakers? Dr. Kaushik wants to interact. You please go on, sir. Please unmute, sir. Please unmute.
Is it okay now?
We can hear.
Yes.
Mine is not a question. I just want to share, because this was a very interesting topic. I happened to work on endosperm genes in rice in 1986 to 89 at the International Rice Research Institute with Dr. Kush. And we had about 15 different mutants which were controlling starch biosynthesis. They were mutants producing 0 to 41% amylose content. You know, amylose to malopectin is an important determinant of grain quality in rice crop. So we studied their expression in indica and japonica backgrounds. You know, indica and japonica background, they are ecologically different types. And to our surprise, these amylose content produced by these mutants, for example, dull mutant in japonica was slightly lower, whereas in indica background, it was slightly higher. Similarly, two mutants were of great interest. One was amylose extender, which we saw in maize recently. This gene, when we transferred from japonica to indica background in IR36, it increased the amylose content from 27-28% to 40%. And when we studied its starch structure under electron microscope, the starch starch granules were irregular in shape.
Okay.
Similarly, the other interesting mutant was sugary, where, in that particular mutant, the phytoglycogen were not converted into starch. We could not find any starch granules. So these were some very interesting, and then we located the different genes or different chromosomes. At that time, trisomic analysis we used. And we found that there are 5 different non-allelic genes controlling low amylose content, the gene controlling sugary and amylose extender. So we were able to look at about 5 genes or different chromosomes using trisomic analysis. What I wanted to share on rice. Thank you.
Thank you.
Thanks. No, it was really interesting. It's in work. Yeah. So still people try to understand how you can influence the amylose content. Yeah. Of course, you can knock down GBSS1 today. Yeah. So then you miss starch. You miss amylose in the starch. There's also some other proteins known to influence the targeting of GBSS1 to the starch granule. And so if you they can have GBSS1 but miss this targeting protein, and then they have also no amylose. So this is still similar under the debate until today, how to influence the amylopectin amount. And as I mentioned before, very interested in this regard is also still, where is it? Where is amylose? Because we don't know where it is. There are also some ideas that amylose may be just something like a not totally synthesized amylopectin molecule. So that is weakly branched, and so it looks different to a typical amylopectin molecule. It is still a very interesting topic. So and it has a huge impact for applications, right?
Thank you. Thank you for elaborating.
Anyone else is interested to interact with the speakers?
Students would like to interact?
So maybe I lost because of our English.
Thing. Chat box. What question only?
It's very difficult. So there is one question in the chat box. Suvarna, can you please interact?
Is the amylose and amylose
Because it's madam, it is.
same, very same in two different rice varieties?
Dr. York, there's one question from Dr. Suvarna. If the amylose and amylopectin content were same in two different
Glycemic
rice varieties, will the GI be same? Will it influence the glycemic index? That's her question.
To be honest, this question is open. So amylose and amylopectin content, is that the same in two different rice varieties, means only that the proportion to each other is the same. So, and of course, if you have a different surface area, you have a different possibly interacting surface for enzymes to act on it. Yeah, and that's why you have to look to additionally parameters to judge. Yeah, if the size and the morphology and so on is the same, and also the inner structure is the same, I would say yes. But just on these two parameters, I would say I cannot say anything, because it just reflects the ratio.
I have one question. You were talking that starch is not genetically determined but metabolically regulated. Can you throw some more light on that? Because you're working on mutants. So what do you mean by not genetically determined and metabolically regulated?
Okay, what I meant by this is the parameters I mentioned. The parameters as surface, number, and so on cannot be genetically determined, as you can see in the double mutants, where these two enzymatic activities are missing. You can find situations where you had a lot of granules, at big granules, and in other situation you have only one granule or no granule. Yeah? So that means this parameter, like granule number and morphology, cannot be strictly only genetic regulated. Of course, genetic regulation exists in this case when you knock out, for example, the first enzyme starting starch initiation. So, for example, if you block the ADP glucose pathway, yeah, then no starch can form at all, and then all the later regulating metabolic regulation cannot change anything else. But in principle, these are affected mainly metabolically. But that is a good question, yeah, because we have to keep that in mind always, yeah. All sugar, so every sugar pathways are not genetically determined. As you can easily think about N-glycans, yeah, N-glycans in the liver and N-glycans in the brain is totally different. And you know, for example, if you have cancer, then you change also the N-glucosylation of proteins. Yeah? And there is no genetic reason for it. It's metabolic alteration. Yeah?
So if there are no questions, please check in the chat box again. I think we'll come to the last leg of this session, that is the formal vote of thanks. So on behalf of Indian Institute of Rice Research, members of the seminar committee, and on my personal behalf, I profusely thank Dr. Habil Joerg Fettke for delivering this talk on new aspects of starch metabolism insights by looking closely on his maiden visit to India. Sir, it was a wonderful talk going deep into the biochemistry. Yeah, we really enjoyed your talk. I also thank Dr. Julia for enlightening us on starch phosphorylation, impact challenges and analytics, and application of the phosphoesterification for industrial uses. I extend my heartfelt thanks to all the dignitaries, life members of SARR, and scientists and student fraternity, and our valuable ECRIP cooperators for their active participation both online and offline, ensuing fruitful discussions. I also thank our AKMU head, Dr. Sailaja, for arranging all the logistics for streaming of this webinar. Thank you, one and all. Thank you.
I thank again all for listening us.
Thank you.
Thanks.
Yeah, this meeting has come to an end. We'll close it down. Thank you, one and all, for the active participation. Thank you.
Can we close it?
Okay.
Oh, that was